Good morning and welcome to Enjoy Science. This is a series of lectures given by the MD Anderson faculty members to share and enjoy the research advances ongoing at our institution. It is organized by the Office of the Chief Scientific Officer, Dr. Dreita, and it is open to the scientific community here and around the world. I'm Alessandra Ferraioli, and I have the pleasure to assist Dr. George Kalin in coordinating these lectures. The presenter today is Dr. Koichi Takahashi. Dr. Takahashi is an associate professor in the Department of Leukemia and Department of Genomic Medicine, Division of Cancer Medicine, the University of Texas MD Anderson Cancer Center, Houston. Dr. Takahashi obtained his MD from Niigata University Faculty of Medicine in Niigata and his PhD from Kyoto University, both in Japan. In 2008, he entered the clinical residency in internal medicine at Beth Israel Medical Center in New York, followed by clinical fellowship in hematology oncology here at the University of Texas MD Anderson Cancer Center. After he completed his fellowship in 2014, he joined the Department of Leukemia, and soon after, in 2015, the Department of Genomic Medicine as a clinician scientist. Dr. Takahashi has received multiple recognition and awards during his training and academic career. Most recently, in 2020, he was nominated the Andrew Sabin Family Fellow and received the Junior Faculty Scholar Award in Basic Translational Research by the American Society of Hematology. He has numerous publications in high-impact journals, and both during his training and in the early part of his career. He is a well-known expert in the field of clonal hematopoiesis and genomic characterization of pre-leukemic and leukemic conditions. Today, he will be presenting on the topic clonal evolution and heterogeneity of acute myeloid leukemia. Before I give the microphone to Dr. Takahashi, I would like to remind everyone to use the chat for questions and comments. Koichi, please. Thank you.
Thank you very much, Dr. Ferraioli, for a kind introduction, and thank you for inviting for this Enjoy Science seminar. It's really a pleasure to present our work and also others regarding this clonal heterogeneity and evolution in acute myeloid leukemia. This is my disclosures, and none of these relationship is relevant to today's topic. So in cancer treatment, there are two types of heterogeneity that we care about. One is the inter-tumor heterogeneity, or in other words, inter-patient heterogeneity. So that each patient has different genetic profiles or different phenotypic profile of their cancer, and therefore one-fits-all size approach sometimes just doesn't work. You need to tailor your treatment based on the genetic or phenotypic profiles of your patient. This is the inter-patient or inter-tumor heterogeneity. And because of the advancement of genetic sequencing technologies, this genetic diversity across the cancer patients are becoming enormous, especially in acute myeloid leukemia. And now there is about almost like 117 cancer driver genes have been identified in play for acute myeloid leukemia, and your patient, the patient in front of you, can have a different combination and different patterns of those genetic mutations. And these are actually the challenge for the treatment of acute myeloid leukemia, but at the same time, it's offering a lot of opportunities to develop molecularly targeted agents against your patient profiles. So we now have various types of inhibitors like FLT3 inhibitors, IDH1 inhibitor, IDH2 inhibitors, that is going to target your patients who have these genetic abnormalities. What today I'm going to focus on is intratumor heterogeneity. So the heterogeneity across each leukemia cells that your patient within the one individual patient. So your patient can have multiple genetic mutations, and when I talk about genetic mutation today, mostly it's a somatic mutation and not the germline mutation. But your patient can have a multiple combination of the somatic cancer gene mutations, and although when you take bone marrow aspiration and when you see the samples under the microscope and all the cells looks like they are same, just they look like just same leukemia cells, actually their genetic or phenotypic or epigenetic or transcriptomic profiles are different among each individual leukemia cells. And that is this intratumor heterogeneity, or sometimes we call it clonal heterogeneity. And again, that heterogeneity could be genetic difference or epigenetic difference. And also, we paid too little attention in the leukemia field about the spatial heterogeneity, but this difference in where your cells locate also matters tremendously, especially in the solid tumor fields. And I think it is also going to be important factors for the liquid tumor or the leukemia as well. And why do we want to really study this intratumor heterogeneity? Why do we need to understand the diversity among different cancer cells or the leukemia cells? There are both biological and clinical implications by understanding that. If you describe intratumor heterogeneity well, you actually have an opportunity to actually understand the evolutionary history of your tumor development. Essentially, the heterogeneity or diversity is a consequence or the result of the evolution. So if you describe the heterogeneity well, you have an opportunity to understand evolutionary history. So that is one reason. And also the second reason is in the tumor sample, it's actually not only the tumor cells exist. There's a variety of normal cells, or in other words, the microenvironment that feeds these cancer cells or the leukemia cells. So understanding the intratumor heterogeneity also includes understanding total ecosystem of your cancer samples or the leukemia samples. And from clinical side, this intratumor heterogeneity is known to create a challenge in the therapeutic resistance or the recurrence and the relapse of the cancers. So understanding the intratumor heterogeneity helps us to tailor your treatment that will improve the chance of the cure, or also to prevent the resistance and the relapse and the recurrence. So for both reasons, I think understanding the intratumor heterogeneity is very, very important in the cancer research these days. Today, due to the interest of time, my talk would mostly focus on how to describe the clonal architecture and how to infer the evolutionary history of the tumor based on that clonal architecture information. And also, I will touch a little bit on the relationship between the intratumor heterogeneity and the therapy resistance. I cannot spend enough time on talking about the tumor ecosystem microenvironment part, but again, there's a bunch of interesting publications around there, and in the future time, maybe I'm able to go over those too. So let's start from the clonal architecture and evolution in AML. Again, you have your patient in front of you who is 52-year-old male with acute myeloid leukemia. Their chromosome looks normal, but when you do a genetic sequencing, you detect 4 types of cancer driver mutations. For example, DNMT3A, NPM1, IDH2, and FLT3-ITD. These are the most common genetic somatic gene mutation that we often find in acute myeloid leukemia patients. And when we see these type of patients in the clinic, the immediate questions that physicians will have is, well, we detected 4 cancer gene mutation in this patient, but do these mutations co-occur in the same cells? Even though it is co-occurring in your patient, do these mutations really co-occur also in the same leukemia cells? Or based on these information, how can we tell about how this AML had developed? Did it start from acquiring this DNMT3A mutation because their variant allele frequency is the highest, and followed by NPM1? And then what? did this patient acquire IDH2 mutation after that, or simultaneously acquire IDH2 or FLT3 mutation? Those questions arise when you see these informations, and understanding the clonal architecture can definitely help answering those types of questions. And much of the work in trying to understand a clonal architecture have been relying on the use of this next generation sequencing of the bulk samples. So when you have a patient with acute myeloid leukemia or any sorts of cancers, first you get a biopsy of that specimens, and then you smash that samples and extract DNA out from it. And then you put those mixture of the DNA from the aggregate of the cells into the next generation sequencing. And you obtain these genetic mutations along with this variant allele frequency measures, how many reads that these mutations are in those genes. And this process is often referred as the smoothie theory, that you have lots of fruits in front of you, and essentially what we are doing is you're blending all these samples together and creating a smoothies. And later on, you're trying to identify the proportion of the ingredients inside your smoothies. And there's a variety of computational algorithm that helps identifying this proportion of these ingredients and reconstructing or deconvoluting the clonal architecture based on this bulk sequencing samples, such as the PyClone, Cyclone, and Expand, and there's other variety of algorithms. And these algorithms do excellent job in trying to identify how many subclones or number of clones that your sample have, and also the hierarchical structures of these mutations. However, since when you use these bulk sequencing, when you extract the DNA or when you smash your samples, essentially you're losing the cellular identity information. So no matter how the computational algorithm is sophisticated, it is almost impossible to reconstruct every pieces of the clonal architecture, or often called the subclonal architecture. So here we are showing some examples of that in the right side here, that when you have this type of genetic mutation with the variant allele frequency information, your algorithm will try to cluster each mutation based on their variant allele frequency similarity and create the clonal architecture. However, with the bulk sequencing information, since you have already blended every cells and lost the cellular identity information, you are not able to determine whether the last subclone mutations are in the branching structures or in the linear structures. This job is extremely difficult to tease out if you only have the bulk sequencing samples, especially with the one-time samples. Sometimes when you have a multi-time point samples or multi-location samples, this job gets a little bit easier. However, if you only have a one-time sample, one-time point samples, this job is often very, very difficult. So the idea was that if that is so difficult, why not just sequence each individual cells one by one and analyze the genetic information of individual cells one at a time. And the concept is very simple, but the feasibility of that or making this happen has been very, very challenging. Nonetheless, multiple investigators in the past have tried to perform this almost like in manual fashion. So essentially sorting each single cell using the flow cytometry and putting each single cell into the 96 well plates and sequence each single cell inside those 96 well plate. But because these technology, there are several limitations on this single cell sequencing that has really hindered this method to become ubiquitous. First, again, this isolation process using the flow cytometry is very labor intensive. And because of that, it's inevitably low throughput. So all of these historical studies have profiled a single cell genome in a scale of 2-digit or maybe 3 digits, or the maximum hundreds or few hundred was almost the maximum number of cells that you were able to interrogate. And after you isolate the single cell into a 96-well plate, then we need to sequence them, but the absolute amount of DNA in single cell is just too little to directly apply that to the sequencer. So there is this whole genome amplification process that is necessary to amplify the amount of DNA in order to run these samples to the sequencer. But this whole genome amplification has been associated with introducing many false positive mutations, or also the false negatives that is called allele dropout. And variety of whole genome application technique has been published with false positives and the false negatives. And this is reviewed excellently by Nick Navin in this molecular cell review. And also for the single cell DNA sequencing, there is a unique problem of accessibility, that DNA is compacted inside the nucleus and in the chromatin, so very difficult to access those. Compared to RNA, which they are in, vast majority of them are in the cytoplasms. So because of this unique limitation in the single cell DNA sequencing field, that this low amount and also the difficult accessibility issue, it was no surprise that in the single cell field, that in the RNA sequencing was much more easier, technically easier, and get commercialized fast or earlier compared to the single cell DNA sequencing platform. Again, this is a figure from the Nick Navin's review in 2015, going over the history of single cell sequencing technologies. But the single cell RNA sequencing was commercialized much earlier than the single cell DNA sequencing platform. In 2018, or actually starting from the 2016, we were very fortunate to collaborate with the company in San Francisco called Mission Bio, and we were able to help in the development of this high throughput single cell targeted DNA sequencing technology. Now is commercialized and called Tapestry. This platform revolutionized in a way to automate this single cell sorting or isolation process, as well as the targeted DNA sequencing of the single cells. And the first focus was in acute myeloid leukemia, so we were lucky because we were investigating that. Compared to the historical methods, this platform really improved the cellular throughput. So with the one run, we were able to genotype roughly 5,000 to now about 10,000 cells in one run. And compared to the historical technology where we were genotyping like roughly 50 or 100 cells, this was significant improvement in terms of the cellular throughputness. And also, this does not involve the whole genome amplification process and only using the targeted amplicon amplification. And therefore, allele dropout and also the coverage issue was much lesser compared to this whole genome amplification involved technologies. We published this technical principles and also the feasibility of doing this in the real AML patient samples back in 2018 in the Genome Research. And then now last year, there are multiple publications using these technologies to single cell genotype large amounts of AML patient samples. The work here, since I'm going to take a liberty of presenting our own work, but those multiple papers published, and all of these papers more or less have shown the same results, which also supports the reproducibility of these platforms. So I would encourage reading other groups' paper. But here in today's presentation, mainly I'm going to focus on our work that was published in this Nature Communications. So in our group, Dr. Morita, who was at that time a graduate student in my lab, took this platform and essentially performed this unbiased, large-scale, high-throughput single-cell genotyping of AML, real AML patient samples. And in our work, we have profiled roughly 150 bone marrow samples collected from 123 AML patients. And we initially used this 50 amplicons and across the 19 genes panel that was developed by the company in the beginning. And later on, we had created our own customly designed 290 amplicon, 38 genes panel. And since this platform was the kind of the early platform, we wanted to test how this platform is doing. So we concurrently performed the bulk sequencing to orthogonally validate these genetic mutations that we detect from the single cell platform, as well as SNP arrays to validate the zygosity of the mutation that were detected from these single cell technologies. Here I am showing some of the QC metrics of this platform. Again, median about 6,000 cells were genotyped using this platform in each run. And in some cases, we were able to genotype 10,000 cells with one run. We achieved about 48x per amplicon per cell coverage, which we think is deep enough to genotype the cell, each single cells reliably, because there's technically two copies of DNA in each single cell. And what we were also assured to see was that this good concordance between the varying allele frequency of bulk sequencing versus the single cell DNA sequencing technology. This means that although you are sequencing only 6,000 or 10,000 cells from your sample, but it is a good representation of the total bulk samples that you are analyzing. And also, when we compare the mutational detection between the bulk sequencing and the single cell DNA sequencing, essentially 100%, close to 100% of the mutation that we detected by the single cell DNA sequencing were also detected concurrently by the bulk sequencing. Or in some cases where we couldn't detect by the bulk sequencing, we were able to validate those mutations by ddPCR. So the mutation detection, the variant allele frequency concordance was excellent with this platform. We also tested what is the limit of detection using this platform, which was about 0.01% of the cellular population, and the estimated allele dropout based on the commonly occurring SNPs were around 5.8%. And when you perform the single cell genotyping of your samples, you get this type of clonal architecture heat map for each individual patient. For here, each cell is lined up as a column and each mutation is listed as a row. And this is the scale of 1,000 cells. So in this sample, we were able to genotype roughly 5,000 cells from their samples. And when your gene of interest is mutated, it is listed as a blue. And if it is wild type, it is white. And if we could not determine the genotype based on, for example, maybe low coverage of this low coverage or for a variety of reasons, then it is described in a gray scale here. And when we were looking at these heat maps for each cases, we started to observe this interesting co-occurrence or the mutual exclusivity at the cellular level. So for example, in this patient who had a multiple RAS mutation as well as FLT3 mutation, each of these RAS mutation or FLT3 mutations were segregated or staggered at the cellular level. So they were mutually exclusive and they were not co-occurring in the same cell. The similar phenomenon was also observed in the second case where this KIT mutation and the FLT3 mutation are also mutually exclusive to each other at the cellular level. In the third case, where we detected both IDH1 and IDH2 mutation, different homologs of these genes, these mutations were also staggered at the cellular level and were not co-occurring in the same cells. And in the last case here that I'm showing here, this patient had two different types of FLT3 mutation, one in the tyrosine kinase domain or the internal tandem duplication mutation. And these two different mutation in the same genes was also not co-occurring at the same cellular level. And we were able to see this beautifully with this single cell DNA sequencing. And these mutually exclusive pattern among this functionally redundant oncogenic mutations suggest to us that if you have too much of these functional redundant oncogenic mutations, it might be deleterious or not providing a selected advantage to the cancer cells and therefore either negatively selected or it just never happens. So you only need one oncogenic mutations for your cancer cells or leukemia cells, and you don't really need too much of these genetic mutations. And when we summarize these intracellular level mutation co-occurrence or mutual exclusivity patterns, it was gratifying to see that this was very similar to what we had observed at the patient level. So the patient level mutation co-occurrence or mutual exclusivity was largely confirmatory at the cellular level observations. Although in rare cases, we did find some unique mutual exclusivity patterns or the cellular co-occurrence patterns in the single cell analysis, but overall, the pattern was largely confirmatory among the patient level and the cellular level. So now that we have this detailed single cell level clonal architecture, the next task is to reconstruct the mutational history based on this single cell genotyping data. And why is this important? Why do we need to reconstruct the history of the tumor development based on the clonal architecture? Because the reason is that in acute myeloid leukemia, we just really don't have a good understanding of how your tumor developed from the normal tissue to pre-cancer phase and to the malignant phase. This is in contrast to other solid tumor, especially with the colon cancer, where there is extremely good understanding of how the benign polyps develop into the malignant invasive cancers. In the colon cancer field, there is this very good understanding of phenotypic evolution. And not only that, they also have extremely good understanding of what genetic evolution that will accompany during this phenotypic evolution from the benign polyp to malignant transformation. And in addition, they also have this time scale that this process will take approximately 10 years from the benign polyp to become the malignant cancer. And therefore, based on that, they have developed this preventive strategy to recommend a colonoscopy every 10 years in order to prevent this benign polyp to become the malignant cancer. This type of pre-cancer to cancer development concept is not really well established in acute myeloid leukemia. And for those reasons, we thought it is important to understand the mutational history, or at least evolutionary history of your tumor development using this single cell clonal architecture information. People have done that and try to do that from the bulk sequencing data too. You can actually infer the relative order of the genetic mutation based on the variant allele frequency hierarchy. And the bulk sequencing does a great job on at least getting kind of a snapshot or the good ballpark of what is the mutation order. However, single cell genotyping data will provide much more granular view of this mutational history or the clonal evolution history. In order to reconstruct the history based on the single cell genotype data, we collaborated with ETH Zurich group, Dr. Björn Winkel, and also the Katharina Jan, who had previously developed this site algorithm, which was specifically designed to reconstruct the mutational tree based on the single cell genotyping data. Using their algorithm, we were able to develop or reconstruct the tumor history, one with the linear pattern and also this branching clonal evolution pattern. Among 123 cases that we had, half of them exhibited this linear pattern. On the other half, we detected this branching patterns. And some of this branching clonal evolution pattern showed a very interesting evolutional pattern, like in this case where we have observed this conversion evolution in AML, which means that there is a multiple parallel subclones independently evolving, however, having very similar oncogenic functional mutations. In this case, essentially all of these parallel clones were having a combination of NPM1 plus IDH1 or 2 with the RTK and MAP kinase pathway mutation. This is similar to the conversion evolution pattern that were also detected in other solid tumor through the multi-location site sequencing. In this case, I'm showing the renal cell carcinoma examples. So now that we have built this mutational history, or the mutation order, we wanted to know is there any kind of dominant patterns, or is there any significant pattern that we can observe in AML in terms of the mutation order. First, we summarize what is the initiating mutations in AML, and this was not so much surprising that most of the initiating mutation, the founder mutation in AML, involve the mutation that is most commonly observed in the clonal hematopoiesis, suggesting that most of these AML had started from this clonal hematopoiesis time point. But after you acquire these clonal hematopoiesis mutation, actually what mutation you acquire as the second mutation or the third mutation was vastly heterogeneous in acute myeloid leukemia. Here I am extracting this pattern in the DNMT3A context, but in a 15% of the chance your leukemia or AML will start from DNMT3A mutation, but after that, what mutation you would acquire as a second is a very, very diverse. Although there may be some patterns here, there is 16% chance after you acquire DNMT3A that you might acquire NPM1 mutations. And the second most, or the FLT3 mutation as the second, and also the IDH2 mutation as a second mutation. But there is also other possibilities to acquire second mutation after, in the context of DNMT3A being the first. And when you summarize entire patterns across this 123 AML cases, we observed extremely heterogeneous pattern of mutational acquisition pattern in acute myeloid leukemia. This is in contrast to more like a myeloproliferative neoplasm, where the mutation order pattern is more fixed or more simpler. Right now, our lab's focus is to try to understand the association with this mutational order pattern or the clonal evolution pattern with the clinical outcomes or the clinical features, which we're currently trying to do right now. So far, I have only focused on how to reconstruct the evolutionary history based on the genetic data only. However, these single cell technologies now have a capability to also simultaneously analyze not only the genetic mutation, but also the cell surface marker. This particular platform, Tapestry platform, allows to analyze about 45 cell surface markers along with the genetic mutation. Now this is called a DAP-seq or DNA antibody sequencing. We have applied this DNA antibody sequencing in some of the AML samples that we have analyzed in that paper. And here, for example, I am showing this case where it started with the single mutation of TET2, followed by acquisition of U2AF1, DNMT3A, and NRAS. And along with these genetic evolution, we were able to understand the phenotypic changes along with this genetic evolution. And this single cell mutant cells, only the TET2 mutant cells, were mutated in the BAST multilineage cells, suggesting that this TET2 started as a benign clonal hematopoiesis. And then along with the acquisition of secondary and the third and the fourth mutation, the phenotype gradually shifted toward the myeloid phenotype and eventually to the leukemic blast phenotype. And by looking at the genetic and the phenotypic evolutional pattern, you are able to roughly tell what was the tipping point from the transition from the pre-leukemia to the malignant leukemia. In this case, we believe that malignant transformation happened either at this acquisition of DNMT3A or at the NRAS mutation phase. So understanding not only the genetic evolution, but along with the phenotypic evolution, like in the colon cancer, where you have this great understanding of phenotypic evolution and the genetic evolution accompanies, that will help us to understand what is the transition point from this pre-leukemia to the leukemia evolution. So now that we have a better understanding of the clonal architecture and the evolutionary pattern at the single cell level, what is really the implication of that in the AML treatment? Tim Lay's group at WashU have in 2012 published this remarkable paper that described how these cancer chemotherapy produces the selective bottleneck to this cancer clones and therefore creating or contributing to the relapse clones that was revealed by this multi-longitudinal time point whole exome sequencing. When you have a single cell, when you apply the single cell DNA sequencing to the multi-longitudinal samples, you get much more granular view again for this clonal dynamics in the patients along with during this cancer chemotherapy. I am showing some of the examples of primary refractory AML patients here. So in the clinic, this patient essentially did not respond to the chemotherapy. They started with 80% blast. After the treatment, they were still 80% blast. So in the clinic, you feel like, okay, chemotherapy did not kill any of the leukemia cell. This patient just was refractory to the chemotherapy. But when you analyze at the individual clone level, actually, there is much more complicated dynamics happening in this patient, even though their blast was completely unchanged from the pre-treatment to the post-treatment. So inside this bone marrow, there are actually multiple clones that were effectively treated with your chemotherapy, while some of the clones are simply resistant or being actually selected during this chemotherapy. So in the end, from the pre-treatment to post-treatment, although your bone marrow just looked exactly the same, 80% blast to 80% blast, actually their sample is vastly different from the pre-treatment and post-treatment. There is lots of clinical clonal dynamics that happen in these patients during this primary refractory phase. So performing the single cell DNA sequencing, even in the primary refractory patient, gives us information that what individuals' clones are responding to the treatment, while some clones are becoming selected or being resistant to certain treatment. And this technology is rapidly applied in many targeted, molecularly targeted agents context. For example, I'm showing several papers here which investigated the clonal evolution dynamics under gilteritinib, which is a FLT3 inhibitor, or ivosidenib, which is an IDH1 inhibitor, and also the venetoclax, which is a BCL2 inhibitor. And what we are seeing in this targeted, molecularly targeted agent context is that this selection of receptor tyrosine kinase and RAS-MAP kinase pathway mutant clone, selection of those mutant clones are the predominant feature of the resistance in these targeted agent field. It's not the only selected clones, but we are observing a lot of frequent selection of these RAS or the FLT3 mutant clones during this targeted agents treatment. And focusing only or profiling only with the DNA evolution probably also is not enough or not sufficient to fully understand the drug-resistant mechanism. Here I'm showing the beautiful paper from the Craig Jordan group, where they profile not only the genetic evolution or also the phenotypic evolution or the differentiation state of the cell during the venetoclax, azacitidine plus the venetoclax treatment. And they have found that not only the RAS mutant clones are being selected, but monocyte clones are inherently resistant to this venetoclax treatment. So just profiling the genetic mutation, you may miss or you may not understand fully the genetic, the mechanism of the drug resistant to the venetoclax, but you also need to understand what is the differentiation state or what is the cell state that your cell is based on the phenotype or also the transcriptomic profile. And that fully gives you the understanding of which cells are being resistant to your targeted agent class. And for that job, there are multiple single cell multi-omics technology and capabilities that is emerging. This is really an exciting time for the single cell field because of this capability of single cell multi-omics that can profile both RNA plus the cell surface marker or the DNA plus the cell surface marker, and also this simultaneous analysis of DNA plus RNA in some cases. So just to summarize today's talk, application of emerging single cell technology has really started to reveal remarkable clonal heterogeneity in AML, which enabling the better understanding of mutation order and history. I wanted to note that we are still under estimating the degree of clonal heterogeneity in acute myeloid leukemia because the platform that we use is still the targeted sequencing. We need to broaden the coverage of the genome, and also we need to involve this multi-omics strategy to fully interrogate the heterogeneity of the tumor ecosystem of acute myeloid leukemia. Again, clonal heterogeneity is a major challenge, especially in the targeted therapy in acute myeloid leukemia that I have described in the multiple settings of the targeted therapy in acute myeloid leukemia, but it also offers the opportunities for development of the newer targeted agents in the future. Multi-pronged evaluation of the cell state using these single-cell multi-omics technology is really necessary to fully understand the intratumor heterogeneity and also the mechanism of the drug resistance. With that, I would like to end here, and I would really like to thank my lab members, especially Dr. Morita and Dr. Wang, who have really performed an excellent job performing this single-cell genotyping studies in acute myeloid leukemia, and also wanted to thank the Mission Bio group who have provided the technical assistance performing this experiment, and also the ETH Zurich folks who have performed this mutational history inference using their site algorithms. And we'd like to also acknowledge the funding source, like the Sabin Family Foundation Ash Scholar Award, as well as the Leukemia Spore Award, and also NIH awards as well. With that, I would like to take some questions.
So thank you, Dr. Takahashi, for this beautiful travel through technology and clinical applications. Really, really fascinating talk. So we have some questions. The first one is, how do you marry the multiple cytogenetic changes in a patient to the clonal architecture changes to define the tipping point to the evolution to AML and the development of therapeutic refractoriness?
That's an excellent question. Right now, we actually are together with this ETH Zurich people. We are developing a bioinformatic algorithm which enables to simultaneously build the tree of copy number aberrations as well as this gene point mutations. And that work is close to being submitted. So at least with that work, in a targeted way, we are able to at least, infer the history of chromosomal aberration in a very limited fashion, like chromosome 7, 5, 8 abnormalities, along with the genetic point mutations. However, we do need to enhance this effort in a wider fashion in order to also identify the fusions or rearrangements. In order to do that, we just need to interrogate broader genome area using these platform or the different platform. Right now, at least when I speak about this tapestry platform, this is still a targeted panel platform. There is a limitation of how much genome you can cover. So there is also a limitation of how much chromosome area you can cover with this platform. I believe that the area that you can cover will exponentially increase as the technology improves. And eventually we should be able to also profile multiple cytogenetic abnormalities, including the rearrangements as well.
So an additional question is, why in an AML DNMT3A is common and the other abnormalities, the additional abnormalities are so diverse?
Actually, I only show the DNMT3A as the most the most common first initiating mutation. But actually, there are other mutation that is also close. You can actually start with TET2 mutation. You can also actually start with IDH mutation. You can also start with P53 mutations too. So it's not only the DNMT3A mutation that is the first. It is definitely the DNMT3A is the most common kind of initiating mutation in acute myeloid leukemia from the point mutation perspective, but that's not the only initiating mutation. I wanted to correct that if I made that a little bit misleading.
The question was why do we see this kind of pattern?
Why? Well, I think that's also $1 million question. I think there is a reason to why too. So when you compare the profile of initiating mutation in AML and also the profile in the clonal hematopoiesis, most of them match, but there is also a slight difference. For example, actually, there is a mutation called PPM1D that is fifth most common in general population as a clonal hematopoiesis, but we almost never see that as an initiating mutation in AML. So the question is why the PPM1D can exist as a clonal hematopoiesis, but very rarely or almost never as an initiating mutation in AML. We don't know. Probably they have a capability of expanding as a benign clone. So you can have selected clones as a PPM1D, but they lack probably malignant transformation, and therefore they just don't become acute myeloid leukemia. What is a biological mechanism behind that? I think that is the $1 million question that definitely requires mechanistic investigation.
Thank you. Then there is a question, it's more of a technical question. One of the audience wants to know if our core capable to perform the surface marker phenotype-based scRNA sequencing. Can they custom select surface marker?
Yes, so I am not the core person, so I'm not probably the best person to answer that question precisely, but what I am aware is that there is also a single cell core that is run by Nick Navin, which is secret funded, and I'm pretty sure in that single cell core, they can perform the site-seq, which is this phenotype plus the RNA sequencing. I am not so sure whether our DNA core run by Erica Thompson also are able to perform the site-seq. Our lab definitely can perform the DNA antibody sequencing. So if you are interested in DNA antibody sequencing, I'm very happy to also help for sure. Although we are not a core, so we may not able to promise the time frame.
Now I have another question. Starts with great talk. To what extent does chromatin remodeling, such as methylation, acetylation, impact clonal heterogeneity in AML? And do you intend to do ATAC-seq analysis?
Wow, yeah, I mean, this is also an excellent question. So in general, diversity or heterogeneity or genetic heterogeneity is driven by the two factors. One is mutation rate, and the second is the time. In terms of the mutation rate, I think a lot of things can drive the mutation rate. One could be the, impairment of the DNA repair mechanism or the exogenous factors. But also, the epigenetic factors definitely play a role in the mutation rate. For example, whether chromatin is open or closed, that definitely affects the mutation rate, which actually was shown beautifully by our colleague Kadir in the genomic department of medicine. So I think, yes, chromatin structures, openness of the chromatin, closeness, even the methylation, those things definitely affect the mutation rate of each genomic site, which overall could affect how the intratumor genetic diversity develops. So I am pretty sure they are in play. Whether performing single cell ATAC-seq can really answer to that question, I am not 100% sure, but I think that point definitely is important for sure.
Great. And one more question. Do you find that the initiating mutation drives the types of subsequent mutations? think I know the answer.
Yes, well, that's also an extremely good question. I think the short answer is probably yes. Although in AML, the pattern and the pathway was such diverse, we did see some strong tendency in some mutational acquisition. So after you acquire DNMT3A, yes, it is more likelier to acquire either NPM1 or IDH or the FLT3 than acquiring something else. So I think there is definitely certain forces there. There is certain determinants there that drives what mutation you will acquire next in the context of first mutation. That pattern is definitely more diverse in acute myeloid leukemia, and we are currently trying to work on to identify or extract the statistically significant order or patterns among those. There's an excellent paper in myeloproliferative neoplasm, though, where the mutation order really determines the clinical also features or the outcome, whether you start with JAK2 or whether they start with TET2 or whether they start with the CALR. Those really, initiating mutation really determines lots of clinical outcomes and the features. In AML, I think we're still struggling to really extract that pattern, but I'm pretty sure there is.
Great. So I don't have any more questions. This was a very exciting talk.
Oh, there's one more question.
There is one more? Oh, yeah, it just appeared. Yes. Sorry. Will sequential scDNA-seq in AML patients be able to guide better targeted therapeutics? Example, when HMA FLT3 followed by another targeted therapy for dominant clones after the initial therapy.
Yes, I think so, but I want to be a little bit careful here that yes, single cell DNA sequencing data provides much more granular view of the clonal dynamics, which is scientifically beautiful and interesting and exciting. But whether or not this can affect the clinical decision making more than what we are doing based on the bulk sequencing is a little bit unclear at this point. I think if you have a multi-time point samples, bulk sequencing can do a very good job, maybe if not a better job, decent job of understanding the clonal dynamics and also what is the mechanism of resistance in your treatment. So yes, I mean, from scientifically, we are super excited to see these granular view. But in terms of the clinical decision making at this point, I think the bulk sequencing also does a decent job as well. So I don't want to say that single cell DNA sequencing is a necessary thing for the clinical decision making at this point.
Great, very exciting talk, very exciting discussion. I would like to invite everyone to that did not have a chance to listen to the talk to look at the recording. I would like to thank you, Liz Kent and Emily Lang, for their assistance. And this is the flyer for the upcoming Enjoy Science talk that will be on Thursday, October 7th, by Dr. Maitra, that will be discussing emerging molecular targets in pancreatic cancer. So thank you, everyone, and have a good day.
Thank you very much.